you're during the day, if you're on a dig, it's 5 bazillion degrees out and you haven't showered in a week or 2 or 3, and you're in the dirt, but you're up close, like close to some dinosaur bone, and you're exposing it, and you're the first living thing with eyes at least, to see it as you're exposing it, and that's a pretty cool feeling. And then at night, because you go out to these very remote places, there's very little light pollution, and so you get like full, Milky Way or whatever. And especially if you have a telescope, you can look at very distant stars, but because of the vast distances in space, the light that travels down and hits your eye, it was produced in the very deep past. The way that evolution has solved the problem of, well, how do you learn about the world around you? And the question of science is, well, let's learn about the world around us. And so curiosity essentially becomes the founding principle of all science. It also makes sense too. I mean, if you don't care about what the answer to a question is, you're probably not going to rigorously work through the problem.
I'm here today with Dr. Evan Saitta, Research Associate here at the Field Museum of Natural History. Evan, thank you for joining me.
Yeah, thanks for inviting me to talk. I always like talking science. So.
Pleasure to have you. So you study a wide variety of topics, correct?
I'd say so. I like to keep it diverse and interesting. There is a method to the madness, as in one thing usually leads into another, and I just, I don't try to limit myself and say, I don't study that.
Right.
If a question naturally arises, I'll sort of follow it, and that sort of led to a diverse portfolio, I guess.
Well, I'd like to try to get through all of it if we have time.
For sure.
But you would consider yourself a paleontologist, right?
It's probably the most accurate description, yeah. One of my more recent papers had nothing to do with fossils, however, and was a pure evolutionary biology paper.
All right.
So, but yeah, I'd say paleontologist, evolutionary biologist, something along those lines.
Great. So one of your areas of research is sexual selection and reproduction in dinosaurs. What have you learned about the sexual variation in dinosaurs?
More so that it's not so much the question of dinosaurs, it's the question of how we study biological variation, including sexual variation, in the fossil record. And so the further I've gone down that avenue, I've actually gone more and more into sort of just pure statistics, actually. And it sort of stems from sort of the sociology of paleontology. Very early on, proposing differences when you find, one fossil and it's got a large dome on its head, and you find another one and it's got a flatter dome on its head. Originally proposing that might be sexual variation, otherwise known as sexual dimorphism, was fairly common. And then I would say within the last 2 decades probably, there's been pushback on that, sometimes for very good reason, in the sense that very often these early papers didn't really use anything in the way of sort of statistical or quantitative approaches at all. But then we ended up in this situation, at least in my opinion, and other people might disagree, in which for one group of fossil reptiles, dinosaurs, sexual variation and what you might call sort of traditional or typical sort of sexual selection patterns kind of went out of style. And you had all these alternate hypotheses brought in to try to explain this supposed absence of sexual variation or sexual dimorphism. Ideas that these structures, the horns and spikes and frills and plates, were all for just recognizing conspecifics, so recognizing species, an individual of your same species, which has some issues because that very process is sort of inexorably linked to things like mate selection, and then you get back into sexual selection. Or this idea that they had very similar selective pressures within each sex, male and female, and between the two sexes in both directions, such that you had like a mutual sexual selection where both sexes express a structure to similar degrees. My issue with that is, in the modern examples, I think those are relatively rare, that very sort of precise equilibrium. And there are other ways other than mutual sexual selection to get, for example, females to sport armaments is usually a good example. So in modern horned animals, bovids, antelopes, females might have antlers if they're of a large body size and live in open environments because they use them to deter predators, right? So they use them as an armament, as a weapon. Whereas the males might have larger horns, they might have horns that are more robust where the tips point inwards so that they can tussle and wrestle and spar, but they aren't necessarily killing and gouging each other. Whereas the females have sort of thinner, straighter horns that point more outwards. And so you can get a situation where just because both sexes sport structure doesn't mean that you don't have one, sort of sexual selection happening. But two, it doesn't mean that there isn't differences on average. And so I've gotten into a very statistics heavy question of like, especially when your sample sizes are so low, as we have in the fossil record of vertebrates, what sort of statistical evidence should you expect? And so there's been a lot of evidence, or a lot of emphasis, I would say, on demanding what's called statistical significance from a statistical test, basically. So if anybody listening is familiar with p-values, right? And there's been a lot of pushback lately, it's sort of the underdog position, against p-values and this sort of over-reliance on p-values in all of science. It ties very closely into the replication crisis in fields like psychology and medical research. And one of the issues, or one way to sum it up, is that by establishing sort of an arbitrary threshold of what probability you think is significant or not for you to either reject or fail to reject a hypothesis, you end up thinking in very black and white terms. Either an effect is present or it isn't. But if you're talking about something like some continuous variable like the length of horns on males and on females, the idea that a finite population of males and females is going to give you zero difference on average or some difference on average, and framing it in that sense is I think a mistaken way to go about it. And then when you add on the issues with the fossil record and these small sample sizes, you're setting yourself up for never finding statistically significant sexual dimorphism. And I showed that with simulation. So for example, if you take human height, the average height of men is greater than the average height of women. Women are not 0 inches tall, like they have a height. The difference is one of degree, not of presence versus absence. But what's interesting is that the range of variation within each sex is such that the two curves, the two normal distributions of men and women, are close enough that when you sum them together, you don't get two peaks. You get a single unimodal distribution, and that is a factor of the statistics, not even of sample size. So if you had infinite men and women to measure under, well, it probably varies according to different populations on Earth, but a typical magnitude of sexual dimorphism in human height would not give you sort of a bimodal peak, which is one of the approaches that people do, is there a statistically significant bimodality to the data? And then, of course, if you bring those down to sample sizes of the fossil record, you're just never going to find it. And so instead, what I try to do is focus on estimating the magnitude of sexual variation and then trying to come up with a way to constrain your uncertainty around that estimate. And so the goal is not, is this dinosaur sexually dimorphic or not? It is, what is your estimate of sexual variation in this dinosaur? And what is the degrees of uncertainty? So for example, your error bars around that estimate. And how does that compare to other species or other data sets? And it allows you to do this in this sort of comparative framework, which is very amenable to the sort of the way we think about a lot of paleontology, where we put things on evolutionary trees and we compare them across species. And so, it's a lot of the sort of debate and pushback around sexual variation in dinosaurs really drove me more towards the statistics, because I was basically sitting there wondering why it was such a controversial thing to say, when there are, of course, many species that, if they have big elaborate structures, the plates of stegosaurs, the horns of animals, those are very often sexually selected, used in intraspecific sexual display or combat, and very often have differences on average between the males and the females. So dinosaurs are an interesting example because we keep running into this question of, we don't find a Triceratops without horns. We don't find a Stegosaurus without plates. But if we draw an analogy to, say, like modern herbivores with horns, armaments, very often the females do have horns as well, and it's for other naturally selected things like predation. Of course, these dinosaurs were not fast. They were like elephant-sized animals, and then there were also gigantic predators that were eating them. And so the idea that we don't find an ankylosaur without armor is not really particularly surprising. They would have lived in environments and based on their very nature would have been prone to things like predation. And so even if the clubs of ankylosaurs were used for combat, related to establishing mates, it's not surprising that the females, for example, would also have clubs if they're at risk of predation. So I think with dinosaurs, there's sort of a methodological hiccup that people we're trying to work through, but also just the fact that they are large and there were predators that were the size of T-Rex,
Yeah.
So it's not surprising to me.
So with the Stegosaur plates, what is the difference between the male's plates versus the female's plates?
It depends on who you ask. If you ask some of the people who disagree with me, they say nothing. But you're talking about a paper I published in 2015. So that was based off of undergrad research I did when I was at Princeton. At the time, a bit of an annoying check here. At the time, it was synonymized, this particular dinosaur was synonymized as a species within the genus Stegosaurus. So it was Stegosaurus musi. It was originally Hesperosaurus musi, and more recently has been split again. So it's always been viewed as a standalone, unique species, but whether we call it Hesperosaurus or Stegosaurus has sort of changed. So at the time, it was called Stegosaurus. I refer to it, I guess, now as Hesperosaurus. I'm fine with that. But what I'm seeing in those plates is variation between, and you see this, it increases, the variation increases with the larger size plates, which could be both a factor of some degree of growth, which you expect the magnitude of potential sexual variation to increase when you hit sexual maturity. Although actually, based on my particular data set, most of those, if not all of them, are at least subadult, probably. And so most of that variation is probably distinguishing between the small plates on the neck and at the very tip of the tail versus the very big ones over the hips and the back and the base of the tail. But again, though, if this is a display structure, it's not really surprising that if you were to have sexual variation, that magnitude of sexual variation would be most expressed in those larger plates. And the particular nature is that you had what was thought of as the very stereotypical Hesperosaurus plates, which were these broad oval structures, sort of like that. So the base of the plate would be here. But what we were also finding is that, well, I guess I only have I'm the only author on that. So I guess what I was finding was that you also had fairly large plates, not as large in surface area. I think the difference was 45% difference in surface area, but quite large plates that were pretty radically different. They were tall, narrow, they had a much more point to them. And you can sort of trace a lot of differences from head to tail, where you have what appear to be these sort of more taller, better defined tips versus these rounder, more wider plates. And I did a variety of analyses to try to eliminate other possibilities. So you look at the bone under thin section to look at the bone tissue to see if it looks like it's actively growing quickly or if it's slowing down or if it stopped growing. In some of the largest plates and some of the largest specimens, you see that they're done growing based on this bone tissue. Sort of like counting tree rings. If the rings are all sort of stacked up right on top of each other at the very edge, that usually means that it's sort of reached that asymptote of growth. So we saw that. So, I didn't think that the tall plates were about to grow into the wide plates or vice versa. Again, there's reasons to think that it's not variation from head to tail for a couple of reasons. There's reason to think that it's not differences in species, because sometimes you'll find bone beds of multiple individuals. There's 2 examples of this, where both plate morphs will be found together. So you can say, well, that probably means that both plate morphs are coexisting in the same time and place. And what we don't see are very radical differences in the rest of the skeleton. Most of the really prominent variation, at least, seemingly so, is in these structures that many scientists will agree are most likely for display. So you go through all these different hypotheses, and if you can sort of build evidence against all the others and say, okay, for now I'm willing to reject those, then you're sort of left behind with sexual variation. And so to draw an analogy with some of these like large modern herbivores, you have these one sex, presumably, so in the sort of standard case it would be the males, but there are species where it's reversed, where the males have these much larger, broader plates, sort of a billboard effect, and then the other sex has narrower, pricklier plates with well-defined apex. The sort of thing that if you didn't want to be attacked by a Allosaurus, meat-eating dinosaur, you might have that. But again, it's not like one sex appears to lack plates entirely. And what's interesting is that if you look at Stegosaurus proper, so similar dinosaurs, you see that in a lot of the larger bodied species, they have plates that have these really like spine-like projections. Things that probably would not look very appetizing to a theropod dinosaur. Whereas some of the smaller species, they're different than Hesperosaurus. They have these sort of more, sub-rectangular plates, but they don't have these long spine-like processes at the tip in the smaller bodied species. But the number of species of Stegosaurus proper is still under debate. I would say most recently there's been a lot of lumping, so that there's kind of one recognized species, but I think there's probably more, because you get some things that are just absolutely giant and some that are fully grown and they're not that much bigger than some of these Hesperosaurus. So I think we're underestimating diversity in the species, and I think that's probably a common trend within paleontology, is underestimating biodiversity of the past.
So do you think the plates of the males are different than the females due to male-male combat or male-male competition?
So the plates probably aren't involved in combat. They're probably displaying. Could be like a Lek-like system too, even. But so the plates are probably selected by the females on the males, if that makes sense. Now, Stegosaurs, both Stegosaurus and Hesperosaurus, they have differentiation of their armor in a way that's very different than other Stegosaur dinosaurs, more primitive Stegosaurs. The more primitive Stegosaurs, they have much more spike-like plates, kind of more or less across the entire body, whereas Stegosaurus retains only the spikes at the very tip of the tail. So it's like these 2 pairs of spikes. And so that structure is called a thagomizer very often. It actually originally derives from a Far Side cartoon. Yeah. And that thagomizer is differentiated from the very broad plates. And so presumably that means there's a functional difference, right? And very often that's ascribed to defense against predators. There's a few meat-eating dinosaur bones with injuries that, you can sort of line up a Stegosaur spike and, maybe. I think that's quite possible. But I do think that we need to be more open to the idea that they might have also been used in intra-specific, so within species combat. So in many species that Lek, for example, even things that have, really fancy feathery displays, males will often fight each other first to establish, say, position within your sort of display arena, whatever you want to call it. And so, it could be that the spikes are being used in combat, and then the plates are being used for display. And then in the females, you have spikes for defense against predation, and then your plates are also sort of more adapted for deterring predators. They're spikier and pricklier. Yeah. It's very hard to say. I mean, you can really only go with how far the fossils go and then draw analogy to modern species. Mm-hmm. So,
all right. Well, let's move on to another one of your topics, feather evolution.
Mm-hmm.
I think most people, when they think of feathers and dinosaurs, they immediately think of Archaeopteryx.
Hmm.
But do we find evidence of flightless birds are from that era?
So that is a very good question, and that inspired my very first research project that I did here at the Field Museum. I came to the Field Museum as a postdoc in 2018, and we actually only relatively recently published that, looking at modern flightless birds and the changes that happen when birds lose flight, right? And it's a very interesting question because even if you look at either extant species alive today or species that are recently extinct, such that we still have their skins preserved, but they're unfortunately no longer with us. You get a wide range. I mean, things like ostriches, they went flightless, off the top of my head, maybe 59, 60 million years ago, something like that. Maybe even longer, actually. It might be more like 62, 63. But versus some of these birds went flightless or lost their ability to fly tens of thousands of years ago, sort of thing. And so you can sort of look at independent losses of flight in an evolutionary sense, right? So you have this evolutionary tree and different parts of the tree lose flight. And then you can see what sort of changes happen over periods of time, right? And that whole project was inspired by the thought that you just raised, which is, flight is lost so many times independently within what's called the crown group birds, so your more recent birds. Presumably, it would also have been lost in the extinct part of the evolutionary tree. And so, I mean, I guess it's not really surprising. It's a lot harder to evolve the ability to fly than it is the ability to not fly, right? And there are certain contexts in which it happens regularly. If you become semi-aquatic, you might lose your ability to fly, the end member of that being penguins. Or if you're flying around and you land on this nice island with no predators, and particularly no nest predators, well, then you might get kind of fat and lazy and, let's start nesting on the ground and let's hang out here. Why are we going? They're on perpetual vacation. And so they lose the ability to fly, sort of thing. So like, the dodo or something might be a good example of that. Islands, lakes, oceans, those have been around for as long as the first volant dinosaur or avian was around. And so I do think it's quite possible that some non-bird dinosaurs who couldn't fly, could not move through the air, they were terrestrial, had ancestors that were, at that point you want to kind of use your term properly, if not volant and able of powered flight, capable of gliding at the very least. I think, so I support what's called the trees down hypothesis, that you had flight evolving after gliding and arboreality, living in trees, rather than sort of evolving in kind of one step from the ground up. And so, yeah, it's very possible that some of these flightless dinosaurs with feathers had ancestors that could, at the very least, glide through the air. So depending on the evolutionary tree that you think is most likely, some evolutionary trees have reconstructed these large raptor dinosaurs, these dromaeosaurs, as being what's called a long evolutionary branch whose sort of root is something more akin to a small, likely tree-living and four-winged gliding dinosaur, where it has these flight feathers on both the arms and the legs. And so if that's the case, then something like Velociraptor might have sort of been the meat-eating dinosaur equivalent of a dodo, basically, where it had an ancestor that was capable of gliding or moving through the air, and then subsequently lost the ability to fly. And our study predicts that if that evolutionary relationship is the true relationship, which we don't know, but you might have similar changes after many millions of years. So you might only subtly change your feathers at first. It doesn't appear that when you lose the ability to fly that the feathers change very rapidly. At the very most, you rapidly change your body size, you get bigger, and your feathers might become more symmetric upon either side of the quill, lose that aerodynamic factor. But it's not until you've been flightless for many millions of years that you get this sort of shaggy ostrich-like appearance and very radical changes. But Yeah, no, I do think sometimes, like, what if Velociraptor was kind of shaggy, like a emu or an ostrich?
Yeah.
And I think it's possible. It certainly is possible. But it's hard to say. I mean, that part of the dinosaur evolutionary tree has just been back and forth all over the place, and there's, you know, a new big discovery that radically changes the relationships every so often. So it's hard to say for sure now, but it's an interesting question.
Yeah. Well, could you talk a little bit about the argument, another one of your papers, looking at surrounding the filaments around bones, are they collagen fibers or are they feathers?
Yeah, so that debate hopefully has been sort of settled.
Has that been put to bed now?
I mean, I think so, and I think a lot of people probably are of the same opinion. Very early on, you had these, this was something I probably could have gotten in before. There's a bit of history that muddies, complicates a lot of these discussions. So when I was talking about this, you know, trees down versus ground up, I've told that to certain dinosaur paleontologists, and they sort of look at me like I have 3 heads, because for a long time, that debate was, say, synonymized with whether or not you thought birds were dinosaurs, right? And so the ground up hypothesis was, no, you have these terrestrial dinosaurs and birds are their descendants. That's the ground up. That's how flight evolved. Versus other people who, this is also something that has been pretty much settled, I would say, who were on the ultimately the losing end of the debate, which was, oh no, birds come from a different lineage of reptile that were arboreal originally, so therefore flight evolved from the trees down. We need to be able to separate hypotheses from each other as things get resolved. So the way flight evolved is sort of not entirely independent, but it is a separate question of, you know, birds deriving from Maniraptoran dinosaurs, right? And in a similar way, the preservation kind of fell into that same thing, where you get these fossils, these amazing fossils from China with preserved feathers surrounding the skeleton, things that are clearly dinosaurs, but they have feathers. Or at least hair-like sort of structures, right? Sort of simple proto-feather structures. And you had the same sort of pushback. Again, I think it was often tied to this, you know, now out-of-date idea that birds were not descendants of dinosaurs. And the alternate hypothesis was, well, no, these aren't feathers. These are the remnants of the collagen fibers from the skin, right? That these were, you know, scaly dinosaurs that didn't evolve into birds. What are you talking about? Look, you know, look at it. You know, can't you see that's a collagen fiber? And that, I think, also several of these debates sort of got tied together. And now I think it's pretty set. I mean, we have fossils now that the feather structures are not these simple hair-like proto-feathers. They are clearly recognizable as bird feathers, right? You know, they got a central shaft, they have the barbs and whatnot. So that debate has largely been settled, but on the chemistry side, this is how I got into a lot of my fossil preservation work, was a lot of this debate about the evolution of feathers. My master's and PhD advisor, my grad school advisor, Jakob Winther, at the University of Bristol in the UK, he was the one who recognized, first in squid fossils, right? He looked at these fossils where you have these squid preserved from these sites of exceptional preservation, and the ink sac is preserved. And people have even, you know, historically, they've used the ink, you know, hey, I'm writing you a letter about the fossils. I'm using the ink from the fossils, you know, isn't that cool? They probably wrote it in proper English, you know, so, you know, much more formal. But it's long been recognized that you get certain fossils with like these color patternings. You have insects that might have like an eye spot, or, you know, feathers with stripes. And what Jakob figured out was that it's the actual melanin pigment that is preserving, and that the molecule itself, it's polymer, is very stable through fossilization. And that opened up a whole sort of subdiscipline of reconstructing the colors of dinosaurs, and a lot of really exciting work. And so with not only these fossils that clearly show that these dark organic stains around these dinosaurs are clearly feathers, not just fibers, right? Combined with the fact that we now have largely worked out the mechanism by which they would preserve, the case is pretty closed, I would say. They're not collagen fibers.
Well, let's move on to another one of your topics: artificial fossilization, taphonomy. Can you explain to us what conditions are necessary to create an artificial fossil?
Well, I guess you got to talk about what we're trying to do, perhaps. So let's start off way at the beginning here. Actually, taphonomy. What does that mean? Well, taphonomy is the study of basically how these fossils preserve. It's all of the processes, biological, chemical, geological, from death to the discovery of a fossil. And so there's certain sort of main phases. You very early on, you'll decay through the action of microbes, through autolytic decay, right? So your enzymes from your own body breaking down your own molecules. So you're rotting. And then, while you're buried, you might get buried deep underground where there's elevated temperature from geothermal heat, there's pressure from the overlying sediments, and you sit there for a really long time. And then maybe you get uplifted and now you're suddenly eroding, and there's oxygen from the air, and there's water from rainwater, and there's erosion, taking you out. And so there's all these processes that go in. And this is something I also want to convey because, I don't want to come across as, we can do it all in the lab, we don't need fossils. Well, you only do the stuff in the lab, the experiments, the simulations, insofar as they allow you to study the fossils. The fossils are what you care about at the end of the day. That's the actual data of Earth history. But the problem is if you look at a fossil, if you look at a really old fossil, if you look at a fossil of something that you aren't quite sure what it was in life, was it a vertebrate? Was it an invertebrate? Did it have these molecules? Did it have those molecules? Was it heavily decayed before? How deeply was it buried? There's all these variables you don't know. And so obviously what you do is you start off by trying to characterize the preservation of the fossil. You'll use microscopy. You'll put it in a fancy electron microscope. You'll use light-based methods, spectroscopy, to look at the chemical bonds. You'll use mass spectrometry to detect the actual fragments of molecules. You do all these things, but you don't have a time machine. You truly can only look at the end result. And if you're lucky, you might have something that's alive today that you can at least compare the very start, right? But as you go further back, you get weirder and weirder stuff that doesn't have necessarily living analogs. And so to sort of connect the dots between what the living thing, or at least your best guess of the living thing, was like, and the fossil, an experimental approach becomes very important. And so if you have results that are consistent, like your overall conclusion is consistent according to data directly from the fossils, data from experiments, and being able to connect the modern tissue to both of those, to the fossils, to the experiments, right? So you might imagine you're characterizing the chemistry of all your samples, and you have the modern sample here, you have the fossil over here, and then you have experiments, and the more intensive your treatment, boom, 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 maybe it becomes closer to the fossil condition. That's a much more powerful argument. And because there's many processes of fossilization, in order to sort of open up this black box of the history, the taphonomic history, you can do all sorts of different experiments. Very common experiments are like just simple decay experiments where you just let things rot in different conditions and you see what things go, what things preserve. You can measure, say, the pH around the carcass or the level of oxygen around the carcass while it's decaying. But I think that only gets you so far. For one, generally speaking, these sites of exceptional preservation, the really best fossils, these like feathered dinosaurs, for example, are usually considered or interpreted to be coming from environments in which decay is very minimal. So for example, they might have been buried very quickly or died really quickly, brought very quickly to an anoxic setting, like the bottom of a lake, right, where you have like a stratified lake and the lower water is is anoxic. There's not a lot of scavengers living there. Aerobic bacteria are not that active. You can still have decay going on through anaerobic bacteria, which can actually lead to interesting sort of mineralization. But in general, the thought is that you're buried with a lot of your soft tissues. I mean, that makes sense. Like, I have a fossil and it has the feathers around it. Well, clearly it had to be buried with the feathers, right? Like, you can take all the feathers off and then put it in the ground, and then, you know, the feathers are preserved. So usually decay is limited, and so then you sort of go along the process, and you're brought into what's called diagenesis, which is just the sort of long-term thermodynamic stability of the molecules over millions and millions of years. And so this is the sort of thing where heating experiments can come into play, and this is something that oil and gas scientists have used over the decades, bioarchaeologists, organic geochemists, and it's a lot of just sort of thermodynamic studies. So you have your starting tissue, your organism, and it's got all sorts of different tissues and biomolecules in it, some of which are very stable through fossilization, some might not be. And by using high heat and pressure, you try to simulate this sort of deep burial over millions of years, but to do it on laboratory timescales. I don't have millions of years to run an experiment, unfortunately. And so you end up with sort of a variety of sort of pressure cooking approaches. And what's really amazing is that alone can really get you very far in terms of, if your goal is to make something that is as close of an approximation to a fossil, boy, it gets you there quite a lot. And that sort of makes sense if your assumption is that these exceptional fossils are not decaying all that much. Then it becomes very much a process, or very much a question of, well, what happens when it's buried underground for millions of years? And it's difficult, and it's not perfect. You know, when you apply the heat and pressure, I'm an advocate for using relatively high-ish temperatures, in the sense that if you expose a carcass or a tissue to the temperature, the exact temperature at which it was buried, I mean, that could be like, I don't know, 50 or 60 degrees, you know, Celsius, maybe even less, 40 degrees Celsius in a lot of places. I mean, the oil window starts at, what, 60 or 80 and goes to like 120, something like that. But if you expose it to a sample to those sorts of temperatures, even like 100 degrees Celsius for, say, a day, you hardly make a dent on it. It does not come out similar to a fossil at all. And that's because of kinetics, right? It's the rate at which chemical reactions happen. So if the thermodynamics is sort of direction, right? Like proteins and DNA, they want to break apart and fragment. That's their sort of thermodynamic tendency. There's a lower energy state if they're broken apart. But the rate at which that happens is also something that needs to be considered. And so you increase the temperature, a lot of chemical reactions happen faster. And so to account for deep time, you really need to run at a hotter temperature. But then you run the risk of exceeding what's called an activation energy of a chemical reaction that doesn't occur at the lower temperatures. And so calibration becomes very important. So you run a series of experiments over a range of temperatures, and then you compare to the fossils. But again, you should be seeing sort of a trajectory of degradation, sort of a, you know, a conceptual decay curve or degradation curve sort of thing. And I took these heating experiments, you know, like I said, a lot of chemists and people who study oil and gas formation have used these things. And I had previously squashed a dead finch, as one does, in sediment, and got it to, you know, sort of look like one of these feathered dinosaurs. And it was really interesting because I was trying to simulate burial underwater. So, you know, we built this rig that would allow for the water to escape out the bottom when you compacted it. We, you know, had it sort of completely submerged, and we were interested in whether the feathers sort of clump together or whether the sediment holds everything in place. Turns out the sediment holds everything in place, and that is good news in terms of interpreting the structure of fossil feathers. But when I was retrieving this finch carcass, I was sat in the University of Bristol fossil prep lab and just using their fossil prep tools, because I had this big block of chalk basically At least I think it was chalk. Big block of chalk that I was just, sort of slowly prepping out, kind of very similar to a fossil. And as I'm looking at this bird, it's like, wow, this works a lot better at making a pseudo rock than I thought. And so very quickly, I thought about these heating experiments, these high pressure, high temperature experiments, which at the time we were using these small sealed metal capsules. And I mean small. I mean, to get a feather in, you had to cut it up and use tweezers to, put it in. And then you seal it off, you weld it shut. And the problem with that is that the components of your tissue that want to break down, sort of the thermodynamically unstable components, all of those breakdown products get trapped. So when you cut that tube open, you're ended up with a smelly feather goo. Sort of smelled like petroleum, burnt hair. It was pretty gross. And all you have to do is look at a fossil feather and you realize that they're not smelly and gooey. So clearly you're trapping things that aren't sticking around. And so I thought, well, okay, what if we just compact a feather in sediment first and then expose it to the same temperature and isostatic pressure. But you're doing it in an open environment, in a more naturalistic environment. And I had gone down to Frankfurt to look at this dinosaur called Stegosaurus, which is this herbivorous dinosaur sort of related to Triceratops. It's got these big quills on its tail, quill-like structures. And we were examining it with laser fluorescence, which is a piece of equipment that my now longtime collaborator, Tom Kay, was custom building and using to study a lot of fossils, and the fossils fluoresce and you can see details. And we were out at lunch one day and I told Tom, I said, well, I had this idea about making, synthetic fossils. And Tom also, kind of like me, likes trying wacky ideas. And he goes, well, what do you need? And I'm like, well, I just want to be able to compact a sample in sediment, this much compactive force. And then I just need to reach these temperatures and these sort of isostatic pressures. And, well, yeah, I can do that. So I went out to his place in Arizona for a week. And we had sort of a Frankenstein, in his garage. He has a full metal workshop in his garage. And at first it was kind of tough going, but by the end of the week we were getting samples that ultimately were publishable. It was really surprising how quickly we got exciting results. Things that really, really look like these exceptional fossils. And so I've been trying to improve upon that and sort of follow that. More recently, Tom and I published what we think is in some way similar to natural copal or amber, where we took tree resin, put it through the same system. And resin's an interesting one because it's the same idea. The unstable components, they break down and they leach through the sediment and are lost, and then the stable components stay behind, and they might cross-link together. And in amber particularly, they polymerize together. And so we've been able to do a wide range of things, and I got people from all different institutions asking to run particular experiments. We just did some stuff on algae, which are of course just as charismatic as T-Rex and dinosaurs. People love algae. But and so it's been really exciting. And I use the term synthetic fossil rather informally and loosely. I mean, they are not identical, no. Because, so one big reason is that we're only simulating one part of the process. And what we're doing now, and actually what I was on the phone with earlier today is a UC Davis scientist who is attempting to run these rotting experiments, decay, and then we're working on a particular mechanism to transfer them over to my rig, where Tom and I pressure cook them, basically. And so I think we'll be able to do some exciting stuff between the two of those, and be able to replicate or simulate a wide range of fossil preservational modes. And again, though, the whole reason of doing, I do it because I think it's cool, but the reason I would give a funding agency is, oh, no, this is very important. This is because, and it is. I mean, your conclusions are stronger if they are consistent with both the fossil and the experimental evidence, right? And you get a better understanding of the underlying mechanisms. If you can make a fossil or something close to it, you might have a good idea of how they form, in other words.
For sure,
Would be the defense of the approach.
Has there been a moment during your field work that has kind of changed the way you see life or time itself?
Yes. I want to give a silly answer first. So the silly answer is I bit through my tongue in Argentina eating steak milanesa, and couldn't speak for 3 days because I had to keep my mouth shut and gargle salt water. We were out in the middle of the Patagonian desert, so I did sort of it's like a silent retreat in the desert. That's the silly answer. Actually, my coworkers on the dig probably appreciated all the silence that resulted from me not being able to talk for 3 days. Yeah, that was bad. That we were considering whether I had to go to the clinic and get stitches. Yeah, so don't bite your tongue in the field. No. There is some really poetic stuff, I would say. The example I like to give, it's not perfect because the actual timing doesn't work out. Pesky thing called physics. But you're during the day, if you're on a dig, it's 5 bazillion degrees out and you haven't showered in a week or 2 or 3, and you're in the dirt, but you're up close to some dinosaur bone, and you're exposing it, and you're the first living thing with eyes at least to see it as you're exposing it, and that's a pretty cool feeling. And then at night, because you go out to these very remote places, there's very little light pollution, so you get like the whole Milky Way or whatever. And especially if you have a telescope, you can look at very distant stars, but because of the vast distances in space, the light that travels down and hits your eye, it was produced in the very deep past. For what I dig, it's actually not quite the same. I think it's like more on the order of tens or hundreds of thousands of years for stars that you can see by eye, whereas these dinosaurs are like tens or hundreds of millions of years, or at least 66 million years old. And so, I guess if you were studying mammoth or something, then it might be a better, but it's kind of fun to think that, this thing that you were interacting with during the day, how distant in time it was. Well, now you're looking at the light that was also distant in time, and that's a pretty cool feeling. And it helps. It's very, very primal. Because you're usually around a campfire by this point. You're in your troop of your small group of people that you've spent way too much time with in the last month or so. Right. You're out in the middle of nowhere. Usually a few beers have been cracked open by that point. Maybe someone comes out with an acoustic guitar. So yeah, no, it's quite fun. To be honest, I view field work almost more as a vacation because a lot of my work is laboratory based. It's more fun than anything else, I would say, because, like I said, important science happens. But I'm saying, for me personally, a lot of my work is not necessarily dependent upon finding the fossil on that particular dig. Right. So,
do you have any field work scheduled?
Well, I was going to go out to Wyoming relatively recently with some field museum people, but scheduling ended up not working out. The plan for the next bit of field work will probably be Green River Formation, Wyoming, with an emphasis on collecting plant material there, which will be exciting. It'll be sort of a different style of digging than I've ever done before. Mostly I've dug in mudstones or sandstones, and these are like finely laminated limestones. So that could be fun. To be honest, when was the last dig I was on? I guess it was also Wyoming, but that was a dinosaur dig. But COVID really put a hamper on a lot of field work, and I feel like things are back to normal now. But so actually I would say a lot of my work lately has been more laboratory based. I haven't been out in the field as much in the last couple of years. So
I noticed on your website you recommend one book in particular. Could you talk about why you recommend this one particular book?
Well, let's see if I can get the full title correct. An Instinct for Truth, Curiosity and the Moral Character of Science by Robert Pennock. It's he's a philosopher of science, actually. I'm going to be brutally honest with you. It's a pretty dry book. It's a long, dry book. It's like academic philosophy. It's not pop philosophy or anything like that. But it's incredibly important, and I agree with like almost all of it. And what's interesting about it, though, is it talks about the values of a scientist and the motivations, and sort of at a level one below what you're often taught. What you're often taught, like, well, how do you do science? You open up the biology high school textbook or whatever, and, first you observe, then you generate a question, then you formulate a hypothesis, and the hypothesis has to be of the form if, then, because, and then you gather data, and we can sort of laugh at that from the perspective of, that's the high school level. But to be honest, even at the professional level, I mean, how is a publication always written? Well, you have to have your introduction first, and then you have your materials and methods, and then you have your results, and then, your discussion and your conclusion, and thou shalt never write a paper that is in anything other than that order. And it becomes formalized in a sense that you need to make sure that what it's formalized on is built on a stable bedrock. And what's really cool is that this book, it takes a philosophy analysis. In particular, it uses Aristotelian, I guess they call it virtue theoretic, is maybe the academic term, but the simplified version of that is any virtue taken to extreme becomes a vice, right? And then it combines it with this broad survey of professional, very successful scientists and what they think of as important, and then sort of comes up with the synthesis to produce this book. And this philosopher clearly is very adept at both science and your sort of standard philosophy, because it starts off with kind of an evolutionary perspective, which I thought was great, which is, even if you're talking about the simplest organisms, you have essentially approach versus avoidance. You detect some sort of chemical in the water, and you either move towards it or you move away from it. Maybe it's food, maybe it's a predator, And then you can go to more, complex organisms, and you look at all sorts of things that are intelligent and have periods of extended childhood, right, and adolescence, where they engage in all sorts of play, right? They're doing all sorts of things. They're running around, and they're curious. And if you think about it, that sort of childhood curiosity, so many professional scientists will say, I just still have that childhood curiosity, that sort of childhood passion. But that sort of mode of learning when you are an adolescent is the way that evolution has solved the problem of, well, how do you learn about the world around you? And the question of science is, well, let's learn about the world around us. And so curiosity essentially becomes the founding principle of all science. It also makes sense too. I mean, if you don't care about what the answer to a question is, you're probably not going to rigorously work through the problem. If you only care about, I don't know, maybe a company is funding you to do research, or maybe you just need this paper published in this one journal in order to get tenure. Maybe you don't care about, what the actual answer is. You care about the secondary things. And so he goes through a whole list of things starting from that. So it's curiosity as the sort of central instinct, into sort of foundational virtues, not, at the level of, well, you need to have a large sample size and you need to be able to replicate results. Those are all very important. That's sort of the statistics level. But even below that, there's things like humility in the face of evidence, right? Like, you need to be willing to update your priors when you face new evidence. You need attentiveness. You need to be able to notice things around you and to observe, but you also need this sort of perseverance, because a lot of work is quite arduous, and it doesn't even have to be 115 degrees Fahrenheit out in the field digging dinosaurs. I mean, some laboratory work. Some of my hardest days have been just in the laboratory, just going through a million samples or doing something real hard, or just it's not working, boom, suddenly it works. And so I really recommend that book. It's a technical book, but it gets at something that is even below the level of what people are typically taught in terms of sort of your scientific values. And I loved that. I thought it was really good.
Yeah, there was that quote there. For science and society to flourish, we need not just a philosophy of science, but a philosophy of the scientist.
Mm-hmm. Mm-hmm.
Yeah.
Yeah, and so it's interesting because he's sort of taking a scientific approach to the question of how to be a scientist, in that he was surveying successful scientists, right? You know, so yeah. Anyways, and so I don't know. I hope I try to live up to some of those ideals.
Yeah. You talked a little bit about some hard days, but what is the hardest challenge that you've had to overcome in your life?
In my life?
Yeah.
Boy, geez, that's a doozy of a question. Well, I'll start off with the positives. How about that?
Sure.
I have an amazing support network, my family, and I have a lot of friends who I can lean on, who have given me tons of opportunities that I wouldn't have. I'm very privileged. You know, I'm a lot luckier, you know. I am a Princeton grad, you know, that's, you know, very privileged and elite. You know.
You have quite the academic pedigree.
Yeah, pinky's out, you know. Yeah, how fancy is that? The hardest challenge, I'll be brutally honest, which is that if you go into academia and you challenge orthodoxies, you get some nastiness on the way. And that's why I started off by saying I have a good support network. And I would give the advice to any young scientist that, you know, if you're talented, if you're eager, if you want to go and jump into the active debates, which I encourage you to do, because that's where the science is most exciting. That's where fresh minds and fresh thinking are the most valuable. There have been studies that, like, you know, anywhere from sort of the natural sciences to mathematics, the sort of hard quantitative stuff, for the vast majority of scientists, their best ideas come in their like 20s, early 30s. And so we can't waste talent of these young scientists. We need to support them. And the hardest bit is there's a lot of politics, and it can get quite ugly. I don't want to go into any more than that, but we've set up a system now that needs to be reformed. We put the highest potential in the worst situations. You know, we should be giving the biggest grants not to the most senior scientists, but to the younger scientists. And we need to give them more stability. We can't say, good luck starting a family. Every 2 years, you're going to have to move cities. Every 2 years, your contract's going to expire. It's a recipe for disaster, and it's not a good way to create a healthy society, as you were saying with that quote from the book. So yeah, it's tough. You know, I would say the hardest bit really has been navigating the politics of academia.
Yeah.
So
it is an unfortunate side to it, isn't it?
But, you know, I sort of ask upon it because I'm drawn to debates. I love the contentious topics. You know, like I said, that's the exciting stuff. But, you know, just got to know what you're getting into, basically. And I would say I was probably a bit naive on that when I first went into some of that stuff. So we can talk about happier, more exciting things than that.
Yeah, we could talk about all of it. Well, let's talk about something that maybe went wrong in the field, but you were able to kind of salvage the trip, or
wrong in the field.
Or improvise to make things work.
I mean, it's kind of, honestly, it's a lot. I mean, any given thing. That's kind of the thing with fieldwork is, and especially if you're digging like large fossils like dinosaurs, I mean, you're never going to be able to predict what's coming. I mean, this even happens in the lab with some complicated experiments. Like, you just got to go and trial and error, and you're going to make errors. Anything from, well, we got to get this bone out, but it's overlaid with another bone, so we can't trench around it in order to jacket it and flip it out. So what do we do? Oh, and there's overbank over here, and our generator just ran out in camp, and oh, it's about to torrential downpour and hail. I mean, I don't even know if I could come up with a singular example. I mean, it's just both in the field and even in the lab, like, you're just going to be faced with constant challenges. And I think one of the issues that young scientists have is they see this and they think, oh, you know, I've made a mistake, I can't do this. No. The making of the mistakes is an indicator that you're in novel territory, right? You're in uncharted waters. And so boy, I'm really trying to think if I could, I want to give you some funny story or some interesting thing, but yeah, I mean, it's just constant, we were in Missouri one time digging, and my entire tent got flooded, and it was freezing cold, and I had to hang all my clothes on this line, and just story after story of that, sort of thing. Injuries can happen in the field. You can bite through your tongue on a Milanese steak in Argentina. It was a good steak too. That's the unfortunate thing.
Argentina has great steak. Could you talk a little bit about how would you say that your work as a scientist, as a paleontologist, has impacted your humanity?
My humanity. Well, I mean, that's a tough one to answer because I've been interested in paleontology and the natural sciences more broadly my entire sentient, self-aware life. And so it's hard to say because I don't have a control, all right? Like I can't measure the delta because I don't have a true control. I've always been thinking about these sorts of questions. How has it impacted my life personally? I've become much more skeptical as a professional science of everything, right? And I try, again, it's this sort of Aristotelian, virtue vice. You want skepticism, but you don't want cynicism, and so I try to become aware of that, challenge my own assumptions. It's made me much more in favor of public funding of science. How about that? That's a tangible example. I mean, I think I do have to say, I mean, in my humanity, the more science I've done, the more I've been thinking about some of these deeper problems, like, what in academia is holding us back in terms of the system, right? The way it is now. So for example, I was talking about how we need to focus on giving younger people bigger grants, right? Something that's more akin to the way that big discoveries happen rather than a much more bureaucratic system of, well, this person has this CV of this long and this person has a CV this, exactly. Yeah. Oh, yeah. I mean, I could give you examples that I'm sure a lot of people you've interviewed, anyone who thinks evolutionarily is, you get an appreciation for nature and conservation. You get an appreciation for your interconnectedness with all life, and that could express itself in, say, views on, the treatment of animals and how we pollute and greenhouse gases and all that stuff. But I don't know, I'm trying to give you a more interesting or unique answer than I'm sure you've probably gotten.
What's a book that you've read recently that's fiction, a book of fiction?
That's a bad question because I have basically stopped reading fiction. I watch a lot of fiction, like TV shows and whatnot. To be honest, I think modern TV shows have, some of them, the best ones, have gotten to the level of fiction books. I could also give you some sort of like defense of that, which is like, it's a more complex medium. Therefore, when it all comes together, that's more of an impressive feat. It usually involves much more people, so there's sort of a collective groupthink involved.
What's an example of a
my favorite TV show is Breaking Bad, Better Call Saul. I love those. Absolutely fantastic. Just every aspect of that is well thought of. But yeah, in terms of reading books, I usually do, to be honest, I've been kind of behind on a lot of things lately. I'd usually do nonfiction, but I do stuff that's outside of paleontology or even evolutionary biology. So my favorite nonfiction book is Marcus du Sautoy, who's a mathematician at Oxford. He wrote a book, don't get too excited now, on prime numbers. But it's actually
prime numbers.
Amazing. It's called The Music of the Primes, I think is what it's called. It it draws upon themes, things that I've been thinking about, like trying to think more fundamentally. Cicadas, what is the number of years, their intervals in which they erupt? Usually prime numbers. Why is that? Well, if you have different species overlapping in geography, prime numbers mean that they will emerge in the same year the fewest number of times across an extended period of time. And so evolution selects for prime numbers as their intervals of year. That is so cool.
Yeah, that is interesting.
And also to get back to things like the funding of science. After the First World War, a lot of the mathematicians, being nerds and whatnot, they were pacifists who wanted to just look at numbers and equations all day long, and they actively chose areas of mathematics that they thought would not have military applications. So they avoided things like calculus and went towards number theory and prime number theory. Well, it turns out secret code and encryption became extremely important in World War II, and is also now the foundation of our entire e-commerce, and that's all based on prime numbers. It's the fundamental being that it's hard to predict when the next prime number is going to be, so it becomes this really hard computational problem. And so it goes to show that the biggest advances in science are often the ones that are least expected. There was a different philosopher, I can't remember who it was, another academic, that was saying that the definition of creativity could be something like an idea that is novel, an idea that is practical, and an idea that is surprising. Keyword, the key factor of that being surprising. And so by definition, the most creative scientific solutions and technologies are the ones that you cannot predict. And so let's say hypothetically you had a large economy. We could call it the United States. And let's also say, hypothetically you had a funding agency that in principle was supposed to be a long-term, stable body of funding, thinking very far into the future, and not necessarily on a 3-year return on investment the way that the private sector, venture capital might think. We could call it the National Science Foundation, hypothetically. Well, if your time frame on which you want to have major scientific advances is long, which would be the public aspect of scientific funding as opposed to the private sector, then you want to fund blue skies research. Not because it makes you feel good and, oh, wow, how interesting, we learned about, this dinosaur or whatever. No. Actually, from a very defensible position, you can say that is how you get the best advances over the long term. It's a factor of time scale, right?
How did where you grew up impact or shape your
for sure
your future?
Well, the boring part of that answer, and I'm sure you've heard this a lot, is basically my entire life I've been interested in science. Mm-hmm. So I can't really give you a reason, because that was before I was a fully self-aware organism. But the big thing, first and foremost, beyond my sort of environment, was my parents, who supported my interests. I was probably very annoying because I asked them to take me to the zoo every weekend, and it's like a 45-minute drive from where our house was. And they supported the interest, and I was maybe only a slightly spoiled kid, but they did have a policy. They did have a policy that the kid asked, "Can I get that toy? Can I get that toy? Can I?" No. But if I asked for a book, the answer was always yes. If I said I would like that book, that was sort of their policy, was they would always get me the book. And so I just read a ton, children's book about science, and obviously it was a lot of dinosaurs and animals, but I had books on all sorts of things, planetary science and astronomy and geology and whatnot. And that really fostered my passion. And I grew up about half a mile away from the ocean in North Florida, Jacksonville area. And I must have been finding my first fossils on the beach, probably 6 or 7. I mean, there's fossil shark teeth there. They're maybe 10 to 12,000 years old or something along those lines. They're subfossils, I would say. They still have collagen in them, by the way. I've used some of those shark's teeth that I found from back home as samples in some of my papers. And so, that fostered it. And then, of course, once you sort of realize, oh, wait a second, these are fossils. And then you get a book about Florida Ice Age fossils. And, oh, here's a fossil horse tooth. Here's a, fossil bison tooth. Here's a alligator scooped. Here's a ground sloth tooth, So you can sort of, but it all gets washed out from the St. Johns River and then washed back up onto the beaches. So you just find isolated teeth and bones and fragmentary stuff. But that really motivated it. And, I did well in school. I tried hard in school. And I think that was really more that I always knew I was interested in paleontology, but I never thought of myself as only being interested in paleontology. What drew me to it was its interdisciplinary aspect. You're studying these things that were alive and they've gone through the bias of fossilization. So it's all this geology. You're thinking about past environments, Earth history, geology work. And then these modern methods of, using chemistry-based approaches, for example, or the high-end computational stuff and thinking about statistics. Even when I was in high school, I thought statistics was very fascinating and I liked statistics. And I liked math to some degree as well, and so I think that all fostered it. But I would say, in large part, it was my family, probably, who've always been very supportive. So, I don't know. That's probably one of the more boring standard answers, I guess. I didn't fight any hard. If you're asking for some, I started from the bottom and whatever. I didn't really. I started from kind of a nice, comfortable spot, and it was just very well supported and very close with my family and whatnot. So I have a loving family.
So there's plenty more we could do, but is there a question that you wish I would have asked that I have not?
No, I mean, you covered a lot of the different things that I studied. You asked a lot of interesting questions. I mean, a question that you could have asked.
Well, what's next for you right now?
Getting National Science Foundation funding. How about that? Working on a few papers, trying to finish up some publications. What I'm trying to do is really improve upon a lot of this experimental stuff. Bigger, better, badder, better. Larger sample sizes, closer approximations, combining decay and pressure cooking, this thermal maturation approach. That's what I'm really trying to do, is to be able to take a modern organism, a modern carcass, and bring it through all the major phases of taphonomy, from decay to sort of maturation, and then even simulating weathering. I'm going to plan on buying a plant growth chamber, which you can make it nice and warm and also humid in there. And so that's going to help simulate some of this surface weathering, where you get water and air interacting with the fossil as they get brought up and exposed. So that's really exciting. I have a paper I'm trying to finish up where we have some preliminary data from fossils, but the concentrations are so low it's very difficult. But we have a lot of promising experimental work that sex hormones, and in this case estrogen, might be very stable in the fossil record. And that opens up one of the key issues from your very first question, which is studying sexual variation in the fossil record. So not only is sample size an issue, but you don't independently have a way to sex an individual fossil. Very rarely you do, I should say. And so if you have a chemical approach, a molecular fossil, like a sex hormone, then you can come up with better estimates of sexual variation in the fossil record. Because right now, what we're sort of limited to is looking at the spread of the data and sort of arbitrarily assigning categories rather than having an independent way to sex the fossils. Now, what I showed in my statistics heavy paper was that actually not knowing the sex, there are ways that you could get around that in many cases, and it's actually not the end of the world. But it would be a lot better. It would be a lot better if we did. A lot of uncertainty would then be eliminated. So that's an exciting thing, is some of this experimental work where estrogen is highly stable through these pressure cooking experiments, essentially, which means that it probably has sort of this inherent thermodynamic stability if it's buried deep underground. The question is it's in such low concentrations by the time you get to just a bone that's, gets buried. So, but it's possible, I think. It's possible.
That's interesting, yeah.
So,
but testosterone is not.
Testosterone we haven't really worked on yet. And that's a factor of, we ordered a testosterone, what's called a standard. So in chemistry, you'll get like a standard, which is a purified form of the compound that you can, run through your gas chromatography mass spectrometer. And okay, this is the signal that it gives right here, Believe it or not, testosterone is an anabolic steroid that is highly regulated, and it's not very easy to just order it and have it delivered. And it was delayed. I was in the UK doing this at my old grad school alma mater at Bristol, and we just didn't get it in time. And so we weren't able, I have run those experiments since, but now I need more funding to analyze the results. So to be determined. Testosterone might be more reactive though. There's some reasons why we think that the actual molecular structure might be more reactive than estrogen, but it's still probably relatively stable, or at least what it might convert into should be stable. So again, sort of, there's theoretical reasons to be optimistic, but it's very early days, so we'll see.
Some exciting stuff on the horizon there.
I think so, yeah. Oh, dinosaur mummies. We got dinosaur mummies on the way. Everyone loves a good dinosaur mummy, where we propose a new method or a new mechanism that produces these mummies, which is very interesting because it's very similar to the sort of preservation you get in very radically different environments. Some of these like really old marine organisms, Precambrian type stuff, they get these like what are often called death masks, where they're either sort of templated in clay, sometimes pyrite or iron oxides, and we're seeing very similar things with the fossils. And there's going to be some debate, I think. There's an alternate hypothesis that I think I'm not sold on yet, but it could be another explanation. But we think what's happening is these dinosaurs are dying in a period of drought. They're sort of desiccating the way a natural mummy, say in arid environments nowadays, like you get these like South American mummies, they're cold and dry. It'd be a lot hotter there, but it would have been dry. And so you get this sort of shrink-wrapped skin, sort of almost leather-like around the carcass. And then when you get the return of this sort of more monsoonal season in the late Cretaceous of North America, they get rapidly buried in these sandstone or sandy river channels. And then what we think is that the microbial decay of the skin attracts this mineral template, this clay that forms around it, because it's external to what the skin would have been. It's not direct replacement of the skin. It's forming on the outside, and based off of the patterns of compaction, it looks like it's forming prior to sort of deep burial compaction of the sediment. So it seems to be early. There's other alternate mechanisms. It could have phosphatized through the action of microbes, and then that phosphate could have been weathered away and replaced with the clay very late, like recently, hundreds of years, tens of years. I'm not sure I put that hypothesis first yet, but I'm open to the idea.
Yeah.
So
cool stuff.
Dinosaur mummies. Everyone loves dinosaur mummies.
That's right. Well, Evan, it's been a pleasure. I appreciate the time.
Yeah, it was a lot of fun.
We'll have to do another one down the road.
I'm happy to talk. So happy to talk science. Should we go to happy hour now?
I think so.